Hello everybody, welcome to our last lecture of the macromolecules unit. Today we're going to have an in-depth conversation about proteins and nucleic acids. If you didn't watch the other lipids and carbohydrates one, I'd suggest maybe going back and watching that one as well. Both of those lectures were pretty dense as far as information goes, so I want you to be able to go back through and pick out those pieces of information. If you're having a lot of trouble with worksheet 2, not the first one we did in class that I stamped, but the second one that was stapled and I said was due on Wednesday. I would definitely suggest going back through and watching both of these podcasts a few times, as they should have all the information that you need, aside from maybe drawing the nucleic acid, but we'll get there maybe. All right. So we started color coding these lectures. Anything in gray is something you have in your macromolecule table already. If you didn't know this information already, though, I would suggest writing it again. Because the more times you write something, the better it's going to stick in your memory. Because now you've done something with this information more than once. Reading is really, really passive. It's not the best way to study. Writing things, explaining things, drawing things are the best ways that you can go. Active study habits. So, we're going to talk about nucleic acids and some basic structure as they all contain these three things. They have a 5-carbon sugar that we call ribose. In our last lecture, we talked about how our monosaccharides can sometimes range from between 4 and 6 carbons. Now we've gotten to one of the ones that have 5 carbons. And this 5-carbon sugar is called ribose, and it's present in all of our nucleic acids that we're going to talk about: DNA, RNA, and ATP. They also all have a nitrogenous base, which is how our ribosomes and mRNA and all those enzymes are able to read our genetic code. We always say that DNA and RNA are the hereditary molecules. They have all that information, they pass it on, but I don't think we've talked about why or how yet. And the way they do it is through these nitrogenous bases that kind of write the language of heredity. And then lastly, they have a phosphate group. They will have anywhere between 1 and 3 phosphate groups, and we use those phosphate groups for energy. For DNA and RNA, they use that energy to polymerize. So going from one nucleotide to a whole long string of DNA and RNA, or RNA, or if you're an ATP, then you have 3. And you break those bonds to create energy for your cell, and that's sort of our energy currency for the body, for all organisms. All right, so a little more color coding. DNA info is in blue, and RNA info is in red. Yes, I do want you to write both of them. The only thing that you may not have to write is in gray. So DNA stores our genetic information that we need for reproduction. I think everyone's heard that at least 4 or 5 times at this point. And it also never leaves the nucleus because it is way too delicate. It has our very specific sequence of nucleotides in a really certain order that contains all our genetic information. And if that gets damaged, then every time you reproduce, that damage gets reproduced. And we sort of start to mutate, or we can form some kind of cancer, or a protein doesn't get created properly anymore. So that's why we want to keep that stuff nice and safe in the nucleus. But RNA is a little more durable. And so we can use RNA to make proteins out of. So RNA has a lot of different forms and a lot of different tasks that it does. And so for each separate task, it has a different mRNA that it's going to use to carry out that task. The first one is to copy DNA used to make proteins. So because DNA can't leave, we make photocopies out of the DNA, and that photocopy is an mRNA. That mRNA is called a messenger RNA because it is the messenger for the information that you need to make the protein so you will not die. Second, we have a tRNA. When that mRNA leaves the nucleus, it heads over to what's called a ribosome. And a ribosome is our protein assembly line. It reads our mRNA instructions and starts to put the correct amino acid on a chain to make that protein. We'll talk about how amino acids are the monomers of proteins. If you don't know that already, they are the monomers of proteins. You definitely need to know that. And so our tRNA is called a transfer RNA. What it does is it brings the right amino acid over to that protein making site, that ribosome, and that ribosome can link it together. Now the ribosome is a really, really interesting piece of machinery. It's made out of both proteins and RNA. It uses RNA to create itself, and there's a little bit of folding helped along by proteins. But RNA is in the ribosome itself, and so we call that rRNA for ribosomal RNA. All right? So a bunch of different functions for RNA, just one for DNA, storing that genetic information. Now, they do have some similarities because they're both nucleic acids, and in fact, their structures are kind of grossly similar. They're both made up of ribose sugars, and that ribose is 5 carbons long, and they are both involved in protein production. That's their whole lives. All they do is they store that info that we need to make the proteins that kind of make us. DNA holds the original instructions, and RNA helps in the production of those proteins. They both have nitrogenous bases that help read or spell out that code for the proteins. 3 of them that are in common are called adenine, guanine, and cytosine. We'll talk about that more when we get to genetics a couple units down the road. But one that's unique to DNA is called thymine. We only have that one in DNA, and uracil is only in RNA. So there's one slight difference on the last base, thymine versus uracil. The other 3 are exactly the same. Alright, so now we get to structure. So remember how I said that ribose is the sugar that we use? So we see this pentagon looking thing over here. Glucose was our hexagon, so now ribose is our pentagon. And on the outside, just like a good sugar should, there are a bunch of hydroxyl groups that is characteristic of sugars on a carbon ring. So the only difference here is in RNA, if you look at carbon number 1 and 2, didn't draw that 2 very well, let's do that again. There is a hydroxyl group on the outside there. And there's a hydroxyl group on carbon number 3 as well. Ooh, that's a terrible 3. That's alright. So, because that's there, RNA is single-stranded. DNA, their structure looks exactly the same. They have that same ribose sugar, OH groups on the outside, except when you get to carbon number 2 now, you only have a hydrogen group there instead of your hydroxyl. DNA stands for deoxyribonucleic acid. That deoxy means you've now lost an oxygen. If you lose that oxygen, you got DNA, and you become double-stranded. D for double, D for deoxy. RNA does the opposite of that. It's single-stranded and has both of those OH groups there. So now moving on to our polymers, which are our examples of polymers are DNA and RNA. I haven't been super clear with that thus far. But when we talk about nucleic acids, our polymers, examples of our polymers are DNA and RNA. There's no specific class name like we have for proteins having polypeptides and carbohydrates having polysaccharides. But when we talk about a polymer of nucleic acids, we are talking about DNA and RNA. So RNA, with both of our oxygens attached to carbons 2 and 3, we have a single-stranded polymer. So if you imagine a long 2x4 with a bunch of nails sticking up out of it, and you took that board and you twisted it. When you twist that board, that's what our structure of RNA looks like. It has a lot of nucleotides sticking up out of a phosphate backbone, and that's how that ribosome will go through and read our genetic code. DNA, we have a double-stranded helix. So we took a ladder and twisted that instead of our 2x4. As you can see, there's different bases color-coded in the center of that DNA molecule. I'm going to write the title of each one underneath. I'll just put R for RNA in red and D for DNA in blue underneath. So now we're looking at DNA. In the center, you see all that color coding in the center, and those are 2 nitrogenous bases coming together and bonding through hydrogen bonds. Now, hydrogen bonds are what keep DNA together. They're not very strong, and so the beauty of that is you're able to unzip your DNA to be able to read it. You only really read one of those strands at a time. That's called the coding strand, and there's another one called the template strand, which is made of a copy out of your coding strand. But we'll talk about that a lot more when we get to genetics and the cell. Just know that DNA is double-stranded. It's held together by hydrogen bonds, and that's the only place hydrogen bonds are going to pop up this unit. So we skipped this in class, so I'm not going to have you responsible for it on the PowerPoint. If you'd like to know how to draw DNA and RNA, I'd be happy to show you. I'll probably move this to our genetics unit so we have a little more concrete idea of what DNA and RNA are and how they differ from each other. Alright, so now we're moved on to proteins. This part is in gray, so what that means is you already have some of this information. But if you notice, the functions that I gave in your macromolecule table are a little bit different than the functions we see for these proteins. They do all of those functions. Proteins just do just about everything, but these are a few different functions they do. Enzymes is the same. I gave that one before because it's really important. Proteins are enzymes. Enzymes are proteins. Some proteins are enzymes. All enzymes are proteins. Enzymes are made of amino acids, making them proteins. You must know that. Say it over 10 times in your head right now. Enzymes are proteins. Pause it. I'll wait. Okay, good. So then the next couple things that they do is channels. Channels are little tunnels in our cell membranes that allow larger molecules to come through them, and they're able to regulate what goes in and out of the cell. And those channels are made out of proteins. We also use them for receptors. In your brain, it's constantly talking to neurons are constantly talking to each other, and the way to do that is they send out signals out into what's called a synapse. And that synapse is waiting with a little protein hand coming up, waiting for that signal, and that hand's going to grab that signal and then do something else. So cells communicate with each other through protein receptors. And then again, most of the things your body does are through proteins. Your whole immune system runs off proteins. Cells keep their shape through proteins. There's a whole bunch of different things they do. Transport proteins, signaling proteins, it goes on and on and on. But I want you to know at least 3 functions for proteins on this test. So pick whatever 3 you want, but you got to know 3. The monomer of a protein is an amino acid. Amino acid, anytime you see the word amino, I want you to associate that with protein. Don't latch on too heavily to the word acid because it pops up in other places like in fatty acids and nucleic acids. It's kind of all over the place. But amino is specific to proteins. So if you see amino, think small protein. It is our monomer or our building block for this class, or this family of molecules. And our polymer is called a polypeptide. Poly means many, and peptide is the type of bond between amino acids. So when you bond amino acids together, you have a peptide bond between them. If you have many peptide bonds, you have a long string of amino acids, you have a polymer of a protein. Some proteins have multiple amino acids, or multiple polypeptides. All proteins have multiple amino acids. But some of them have multiple polypeptides. And those are still considered proteins. The reasons we have a difference in classification between polypeptide and protein is because some proteins have multiple polypeptides. So now we're on to amino acids. Amino acids are our monomer, and they look just like this over here. They have one carbon in the center that I'm going to circle in blue, and to the right they have a carboxyl group that we know how to draw, I hope, and we have an amino group that we also know how to draw, I hope. Those 2 things never change. They are constant, and they form the backbone of your polypeptide. So just like we had with our DNA, with our phosphate background, we have an amino and carboxyl backbone for our proteins. Now down here is our R group. R group, with that red arrow underneath it, sort of arrow. Oh well. That thing is variable. When you see an R in chemistry, it's kind of like seeing an X in algebra, except you don't need to solve for it. That means there can be a bunch of different things that bind there, and there's 20 different R groups. I'm not going to ask you to even think about knowing all 20 of those. That's for college. You'll get there. But I do want you to know that there's 20 different side chains or R groups, and every time you change the R group, you get a new amino acid. And so what I did ask you to do for homework was to draw out your amino acid looking just like this. Don't worry about drawing any of the R groups, just write R for right there. So if you didn't get a chance to do that, definitely practice your amino acid drawings because it's important, I think. Alright, so our last slide we talked about protein structure. Proteins are really complicated when it comes to their shape. Remember that all molecules, their most important function is, or most important characteristic is their shape. It defines their function. And so when we talk about protein shape, there's 4 levels that is influenced with. The first level is called our primary structure. Primary structure is just the order of amino acids. Remember that there's 20 different kinds of amino acids, and so the way you string those together matters a lot. And so we call that primary structure. It's the order in which you string together your amino acids. Secondary structure, I don't want you to worry about too much. It's a little more complicated, but it's how those amino acids interact with each other. They can either form beta pleated sheets, like you see on the left right there, which are kind of like if you took paper and folded it over on itself to make a fan, and then spread it all out, you'd have a bunch of kind of pleated sheets, or sharp little hills and valleys in your paper. And that's what a beta pleated sheet looks like. An alpha helix sort of looks like my hair on a good day, and it's a long spiral down through the center of it, and those are usually used for hydrophobic amino acids because they're trying to hide from water. Third, we have our tertiary structure. Tertiary means third. Welcome to Latin. And that means our polypeptide, that's how our polypeptide folds over itself. So when we take the accumulation of beta pleated sheets and alpha helixes, and those things start to interact with each other, the protein will start to fold in a really specific way. We've seen proteins drawn as kind of like these Pac-Man looking cartoon things in our book, and while those are nice to look at and they're pretty and they're easy to think about, those aren't how proteins really look. What they really look like is over here on the left in our tertiary structure. We have a bunch of amino acids interacting with each other, and they fold up in a very specific way. So if it's an enzyme, you'll have something bind to a really specific spot. If it's a carrier protein, it'll pick something up with a really specific spot. So again, proteins don't look like those nice cartoon shapes. And then lastly, we have quaternary structure, which doesn't occur in all proteins because some proteins just have one polypeptide. But if they do have multiple polypeptides, how those things fold together is called our quaternary structure. All right, so I think that is just about it. Oh, drawing an amino acid. That was your homework. Draw it 5 times. There's no fun little chant for that one, but all you need to do is carbon in the middle. Oops. Carboxyl group on this side. Amino on that side. R group down here. And a little oxygen because carbon likes to have 4 bonds. So I know this was a lot of information really quickly. There's no scared faces in front of me to say, Mr. Hutton, slow down, you're talking a mile a minute. So as always, pause the podcast. Maybe watch the podcast the first time and take notes the second time. Do vice versa if you want. Maybe pause and take notes at the same time and take it an hour to get through it. But don't worry about it. Take your time. This is a great way to study. And I'll see you Tuesday. Best of luck this weekend.